Okay, what do you guys say we wade in and get out of this heat as soon as we can? No, it sounds like a plan. Okay. Let's spend some time now talking about the next higher level of structure of carbohydrates, and these would, of course, be the polysaccharides. And polysaccharides include cellulose. Cellulose is a very important polysaccharide on the face of the earth. It is, in fact, a very important structural component of plant membranes, plant cell membranes. Okay? Cellulose is a polymer of glucose, meaning it contains only glucose. This polymer of glucose, okay, is a series of glucoses linked in beta 1,4 linkages. All right? Meaning the beta configuration of one glucose is linked to carbon number 4 of the other glucose. All right? That linkage turns out to be very, very critical. And I'll explain why in just a minute. But suffice it to say that chemically it is different from the linkage we see in carbohydrates in our body. Our body has a form of glucose known as glycogen, and the most common bond that we find in glycogen is actually glucoses linked in alpha 1,4 configurations. Now, these guys actually have different chemical properties. As I say, plants use it as a structural component of their membranes. It gives strength. Glycogen tends to have a very different kind of a structure. It's not long, polymeric, and sort of helical in nature, but in fact it's sort of a very odd diffuse molecule that I'll show you later. Starch is related. Starch also has alpha 1,4 linkages. But again, I'm going to say some more things about those linkages. There's more than just 1,4 in both of those. Cellulose, however, only has beta 1,4 linkages. That's all it has. Okay? And that is important because if we look at our digestive systems can break down starch. It can break down amylose, which is a component of starch. It can break down glycogen. It can break down all three of those polysaccharides with the alpha 1,4 bonds quite readily. It cannot, however, touch cellulose. So the enzymes that will handle alpha 1,4 bonds will not, underline not, handle beta 1,4 bonds. Okay? It's for this reason that that we think of plants as roughage, because these polysaccharides aren't digested in our digestive systems. They add bulk. They are carried through our digestive systems. They help move things through, as it were. And we don't get the energy out of cellulose that we otherwise would. So if we eat lettuce, which is full of cellulose, we get very, very little in terms of caloric content out of that cellulose because we can't digest it. On the other hand, if I am a ruminant, I'm a cow, for example. Cows have a different stomach setup than we do. They have a 4-chambered stomach, and it's called a rumen. And in these 4-chambered stomachs that cattle have, they have a different arrangement. They have a different setup where they can actually harbor different bacteria than we can have in our stomachs. Our stomachs don't contain the same bacteria, and in the cow, they contain bacteria that can make an enzyme called cellulase. And cellulase can break down cellulose. We don't have this bacterium. We can't break down cellulose. They can. So the reason you see cows out grazing in the pasture is that they are actually getting a lot of energy out of the grass and various plant tissue that they're out there eating that we don't get. Okay? And that's thanks to the bacteria that we find in the rumen that make cellulase. Okay, so it's very subtle structural differences can have profound impacts on the digestibility and our ability to eat some of these compounds. Okay, there we go again. Let's see. If we look at glycogen, and I'll say more about glycogen as we get further in the term, this will happen mostly next week, what we see is that glycogen, in fact, in addition to having alpha 1,4 bonds, occasionally has what we call branches, and the branches are alpha-1,6. So if we think about these two guys on the bottom, we can think of this as a polymer, where we might have dozens of these alpha-1,4, 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 going all the way across the screen. About every 10 residues or so, we see a branch. We see a 1,6. So for example, this guy right here has both an alpha-1,4 and it has an alpha-1,6 linkage. That provides a fork in the road, as it were. Then this alpha-1,6 guy up here will in turn have 1,4, 1,4, 1,4, 1,4. So we only see the 1,6 at the point of the branch. Branching gives glycogen some very, very important characteristics for animals. Okay? One of the reasons that I already talked about how hemoglobin was essential for us being an animal. Another thing that we'll see is that the structure of glycogen also is very important for us to be an animal because it provides us with very quick energy. And starch and amylose and other polysaccharides don't do that. We don't see, for example, glycogen in plants. Okay? We see something that's slightly related but not nearly as complex as glycogen is in plants, and I'll talk about that. And actually that brings to mind, I should have mentioned it up here. So in plants, we have other polysaccharides besides cellulose. So plants also, why do we have polysaccharides in the first place? The reason we have polysaccharides is to store glucose. Glucose is our primary source of quick energy. We store glucose so we can access that energy very quickly, and glycogen's chemical structure, as we will see, facilitates that easy access to the glucose. We store glycogen primarily in our livers, our liver, if we have one, I guess, and in our muscles. Plants also store glucose, and they store it in a couple of forms. We talk about starch, but starch is actually a mixture of different polysaccharides containing glucose. It's a mixture. It's not one compound. Okay? So starch is typically comprised of two different categories of polysaccharides in plants. So starch is a plant polysaccharide. It's a mixture. And it's a mixture of something called amylose, A-M-Y-L-O-S-E. Amylose is a very simple polymer. Everything, alpha-1,4, 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 alpha-1,4. It is the opposite of cellulose, which was everything beta-1,4. So amylose is one component of starch. The other component of starch looks a little bit more like glycogen. It has alpha-1,4s. It has some alpha-1,6 branches, but not nearly as many as glycogen has. So glycogen will have branches about every 10 glucoses. Amylopectin, which is the other one, will only have it, which is the plant equivalent, will only have it about every 30 to 50 glucoses. That makes for a lot less branching than we see in animals, and that branching, as we will see in animals, is very, very important for the quick release of energy. Okay, so starch equals amylose plus amylopectin. Yes, sir? It's called amylopectin, A-M-Y-L-O-P-E-C-T-I-N, amylopectin. So amylopectin is the sort of plant equivalent of glycogen. Okay, well, we're moving right along. We want to talk a little bit about now some modified forms of polysaccharides, and we will see that these guys are fairly important as well. One of these modified forms is known, actually let me just show their structures to you here. Chondroitin 6-sulfate, keratin sulfate, heparin. Heparin's a good anticoagulant. Dermatin sulfate, hyaluronate. Okay, I'm going to talk about hyaluronate. Most of what I'll have to talk about is with hyaluronate. Hyaluronate, of course, is also referred to as hyaluronic acid. And hyaluronic acid is a very important lubricant in your body. Okay, so in every joint or most joints of your body, you have protective lubricant called synovial fluid. Your knees, for example, have synovial fluid in them. They provide a cushioning, very much like oil does for an automobile engine. They provide cushioning and lubrication to make that joint move smoothly and not encounter problems. That means that hyaluronate has to have an interesting chemistry that it actually gives lubricating properties to water. And we think about water and we think, well, water is a lubricant, but it's not a very good one. We don't, for example, put water into our car engine as a way of keeping it lubricated. It doesn't work very well that way. And when we think about it, we compare water and we compare oil, motor oil, for example. Water doesn't have that sort of slippery nature that oil has. Okay? So what hyaluronic acid does is it imparts to the water that it is dissolved in a slippery factor. If you're a bioengineer, you know that synovial fluid, you probably know, I shouldn't say you do know, but you probably know that synovial fluid gives elasticity and lubrication, both properties there. Yes, sir? Does it reduce friction? Yes, it will. Mm-hmm. Yep. So that's what I'm thinking of is what I'm calling the slippery factor. But yeah, it allows those ends of the joints to literally move over each other without problem. So there is a reducing friction component. There's also, as I say, an elasticity component, so that I can do this, okay, and it's resisting, okay, the impact of that. So it's actually providing a cushioning that's there. If you have problems with your joints and you have, for example, low amounts of synovial fluid, you will know it, okay? You will know it. Very, very important. Well, the question then arises as to, well, how does, for example, hyaluronic acid give this property? Well, I'll say a word about that in just a second. I want to point out some of these other compounds. Some of these other compounds are things that we find in things like snot, okay? We start thinking about the consistency of snot. We start thinking of, well, there's the slippery factor that he's talking about. What chemically is giving these properties? Well, as we look at these, we see that there's a common feature that they have. They all have a repeating unit that is a disaccharide, meaning this disaccharide may be repeated hundreds or even thousands of times. In some cases, maybe even more than that. Hyaluronic acid in synovial fluid has molecular weight on the order of millions. All right? So many thousands of copies of this disaccharide linked, one after the other after the other. The other property that it has is every one of these guys you will see has very negative charges sticking off of them. There's a carboxyl group here. Here's a sulfate. There's another carboxyl sulfate, another sulfate. Okay? There's a sulfate, carboxyl, etc. So when we think about thousands and thousands of these guys arranged in three-dimensional space, we think of what we have made is a polyanionic compound. Polyanionic, meaning that it has many, many, many negative charges arranged in a polymeric form. DNA has that property. Okay? Anybody ever work with a concentrated solution of DNA? Nobody? Maybe not. Okay. If you ever get a very concentrated solution of DNA, you'll find it's very much like snot, and DNA is a polyanionic compound. So the polyanionics actually do this. The polycationics would do it as well. But the polyanionics do this, and I'm going to show you why or how they do that in just a second. Okay? So this thousands and thousands of copies of negative charge over and over and over actually change the chemistry, the chemical properties of the water that they're dissolved in. Okay? There we go. So what do they look like? Okay, well, that's blasted. Okay. That wasn't quite what I wanted. I thought I had a structure for you. All right, let's go back to this and I'll describe it to you. All right? Let's imagine that I have this hyaluronic acid. So I've got thousands and thousands of copies of this same unit over and over and over. Okay? Something that's polyanionic, each of the copies are being held to each other by covalent bonds, so they can't get away from each other first. Second of all, they all hate each other's guts. Negatives repel negatives. So what do? They try to arrange each other as far away apart as they can, and they repel other units away from them as well. So they actually sort of stratify the solution that they're in, and they arrange each other so as to be far enough apart because negative repels negative, etc., etc. They stretch each other out, and it's that stretching each other out that changes the property of water. Okay? It actually physically changes the property of water. It gives that sort of snotty feel to the compounds that I have just described to you. And that becomes important when I consider a slightly related class of compounds called proteoglycans. Okay? Proteoglycans have a protein core. You can see that shown here in green. And they have polyanionic compounds attached to it. So they could have, for example, hyaluronic acid attached out here in brown. Now I want you to look at this structure and notice what's happening in this solution. These polyanionic compounds are trying to get as far away from each other as they can. That's why we see it all spread out. And that is a reflection of what's happening even if we don't have the protein here. So the protein is the green part. There's the polysaccharide component out here. So that negative charge is causing them to repel each other, and that repelling of each other does change the chemistry of what's there. So I've just introduced a second term. I haven't defined it, so I better define it for you. I described what I called glycosaminoglycans. That was the first group of compounds. The glycosaminoglycans do not contain any protein. Their name tells you something about their chemical structure. So let me review that with you again, because I didn't do that before. Glycosaminoglycan. Okay? Glycose meaning that they are derived from sugars, usually glucose. They're derived from sugars. Okay? Amino, meaning that they all have an amine in there, meaning a nitrogen. Notice there's a nitrogen on chondroitin 6-sulfate. There's a nitrogen there. There's a nitrogen there on heparin. There's a nitrogen on dermatan sulfate. And there's a nitrogen over here. Glycose, amino, and then glycan refers to the polymer. So we have units of sugar. We have amino portions of them, and they're linked in a polymer. Glycosaminoglycans. Notice that the name doesn't have anything to do with proteins. Glycosaminoglycans do not have any protein in them. They are polyanionic glucose-related compounds. Okay? If they have a protein associated with them, so let's imagine I took a whole bunch of hyaluronic acids and I attached them to some proteins, I would make what are called proteoglycans. Okay? So this is a depiction of a proteoglycan. Again, these brown guys might be hyaluronic acid, and the green portions being proteins as such. Okay? So the difference between a proteoglycan and a glycosaminoglycan, the proteoglycan contains glycosaminoglycans, but it also contains protein. Okay? And let's see, what was here? And this now schematically is depicting the same thing I was showing you on the last slide there. There's a whole bunch of units of the individual, there's chondroitin sulfates linked on the side of that. We can see them all sticking out there as fibers. And we can imagine that these guys are all going to try to get as far away from each other as they can. And in fact, they do. Okay. All right. So we're moving along fairly rapidly. We get a sort of a sideline for a second because your book brings up UDP glucose. So I want to show you this one. This is a modified form of actually a couple of sugars. UDP glucose is related to the nucleotide UDP. UTP is what's one of the nucleotides used to make RNA. So UDP just simply has 2 phosphates instead of 3. We see that in the cell, UDP is very commonly attached to glucose. And it's attached to glucose for a very important reason. The reason being that we use this to make glycogen. We'll talk about this later when I talk about the synthesis of glycogen, but I want to talk about the chemistry of it before I talk about it as an enzymatic substrate. UDP is a compound that has a decent amount of energy in it. Okay? It has a decent amount of energy in it. The reason that we use UTP, for example, to make nucleotides is the energy of the UTP is used to make the RNA molecule, for example. Okay? Well, when I combine that UDP with a glucose, as we see here, what I've done is I've just created a molecule called UDP glucose that has a lot of energy. This is a very energetic molecule. UDP glucose is a very energetic molecule. It's much more energetic than glucose by itself. Much more energetic than glucose by itself. Yes, sir? What exactly about the molecule makes it more energetic? Good question. It's the proximity of the phosphates to these hydroxyl groups that actually does that. So as I mentioned earlier, when I put the phosphates onto any sugar, I create a higher energy molecule than what I started with. So that phosphate is negatively charged and really actually favors being left away from those hydroxyl groups. Okay? So chemically, this makes for a high energy bond right here. Now that high energy bond turns out to be important because when we make glycogen, we need energy to add the glucose onto the growing glycogen chain. When I make those alpha 1,4 bonds, for example, I need energy to make those bonds. Well, this molecule actually supplies its own energy. This molecule supplies its own energy. It's an example of a class of molecules that we refer to as activated intermediates. Activated intermediates. I'm going to define that for you in a second, but an activated intermediate is what UDP glucose is. Now, an activated intermediate is a molecule that has high energy. As you see here, it's a molecule that has high energy, and it uses some of that energy to donate a part of itself to something else. So it's a molecule that has high energy that uses a part of that energy to donate a portion of itself to something else. In the case of this molecule, UDP glucose, the high energy is used to donate glucose to a growing glycogen chain. So I said we needed energy to make those alpha 1,4 bonds. The energy actually comes from this molecule. So cells synthesize this molecule as a precursor to making glucose. And we'll see other examples where cells make activated intermediates. If you've ever heard of acetyl-CoA, for example, it's an activated intermediate. Its energy of that bond is used to donate a portion of itself to making something else. At the beginning of next term, you'll talk about the synthesis of citric acid in the citric acid cycle. And that happens because acetyl-CoA has high energy and it donates the acetyl group to oxaloacetate to make citrate. You'll see that next term. Okay, so everybody understand what an activated intermediate is? Why they're important? Okay. All right. Now, I come back now and talk about another collection of carbohydrates relative to proteins. But they're not the same as proteoglycans. And students frequently, one of the more common questions I get is, what's the difference between a glycoprotein and a proteoglycan? Okay? It's very simple. Proteoglycans contain glycosaminoglycans. So we're going to see, if we have a proteoglycan, we're going to see chondroitin sulfate. We're going to see hyaluronic acid. We're going to see any of the glycosaminoglycans. Glycoproteins will not have that. Well, what's the glyco part? The glyco part is that glycoproteins will typically have relatively short oligosaccharides. Oligo meaning a few. So instead of having thousands and thousands of polyanionic compounds on them, glycoproteins will have a few sugars attached to them. They will not have that long polymeric polyanionic compound. All right? Examples. Okay? Examples include, for example, the blood group antigens. All right? So these are molecules that we find on the surface of blood cells, and they give rise to the O, the A, and the B blood types that we see. Okay? These molecules are glycoproteins, meaning that they are proteins that are embedded in the surface of blood cells, and attached to those proteins are short oligosaccharides of specific structure. You can see that the difference between an O antigen versus an A antigen versus a B antigen is not very significant. But the immune system sees them very differently. Okay? So it's for that reason we have to be very careful in mixing different blood tests. We see a very common core, for example, in these antigens. There's galactose. That stands for N-acetylglucose. That stands for galactose. And there's fucose, that odd one that I showed you earlier. Okay? The only difference here is the A differs from the O in having this N-acetylgalactose over here, whereas the B has a galactose over here. Okay. And you can, so, and we, of course we have schematic ways of representing these. I'm not going to ask you to draw those or any of that, but you should recognize that glycoproteins are the ways in which blood types arise and differ from each other. Okay. Now, what we see on the screen is the way that the way that the carbohydrate portion of a glycoprotein is attached to the protein. Okay, two different types here. One type, we have what are called N-linked glycoproteins, and the other are what we call O-linked glycoproteins. Okay, the N-linked glycoproteins, you will note, arise from a linkage, okay, through asparagine. So we see asparagine. Here's the protein sequence up here. Here's an asparagine side chain, and the asparagine side chain is linked to N-acetylglucose. So N-acetylglucose would be found in N-linked glycoproteins. There's the linkage. And then the other portion of the glycoprotein would be, of the polysaccharide, the oligosaccharide would be down over here. Okay? But when we see N-linked, we will see it linked through an asparagine as we see right here. O-linked glycoproteins are the other class of glycoproteins that we find, and they are linked through the side chain of serine. Serine, of course, you remember, has a hydroxyl group side chain, and it becomes linked to N-acetylgalactose. There's N-acetylgalactose, and again, the rest of the oligosaccharide will be going out in this direction. In both cases, notice we see N-acetyl. N-acetyl modification is a very common modification that happens. In both cases, it's on carbon number 2 of either the glucose over here or the galactose over here. Now, these two classes of glycoprotein have different cellular origins, and I'll say just a little bit about that because we're going to start talking about the movement and trafficking of materials through the cell. Okay. Oh, don't tell me I lost it again. Nope, there it is. Okay. Now, I like this figure because it shows a single N-linked glycoprotein. Okay? This N-linked glycoprotein, there's that asparagine that we expected to see. There's the linkage to N-acetylglucose. And what we discover is that N-linked glycoproteins have a common core. We see that common core that consists of, and again, I'm not going to ask you to memorize these, but I want you to recognize that there is a common core. This common core is arranged in a sort of a Y-shaped structure, and it really only contains two compounds. The common core contains mannose. It contains N-acetylglucose. Okay? That's really all that we see there. Okay? Now, we see other things out there, and you might imagine, since they're not shown in gray, that they are not common to all N-acetyl glycoproteins, that is, to all N-linked glycoproteins. And these vary from one glycoprotein to the next. The pattern of these, what I call decorations that are out there, kind of like the branches of a Christmas tree, the decorations that are on that common core affect what the protein does, and they affect where the protein moves to in the cell. Now I'm going to spend some time in just a bit talking about how it is that cells decorate a glycoprotein. But again, what you're seeing in this case is a complete glycoprotein. You'll notice there's not thousands and thousands of saccharides on there. Okay? This guy has about, what, maybe 10, a dozen or so sugar residues on there. Schematically, it's shown on the right, for whatever that's worth. If you like schematics better than names, I don't think it really matters. But we have a common core. But we could have another glycoprotein, an end-linked glycoprotein that would have this common core, and then a different arrangement out here. Notice we have alpha 1,2 bonds, we have alpha 1,3 bonds, we have alpha 1,6 bonds, and we could imagine quite a wide variety of structures arising from those various isomeric configurations. Okay, here's a more complicated one. Again, we see the common core. There is the N-acetyl group. There are the N-acetyl groups. There's the mannose groups. We see things coming out down here that's different. We see things going up here that are different. And we start seeing that there can be quite a bit of diversity between many different glycoproteins. But again, they all have that same common core that we saw in the other one. Okay, now, we need to understand a little bit in the cell about the trafficking and the movement of proteins, because when we think about a eukaryotic cells have organelles. And organelles all have membranes. And so when we start moving things across cell membranes or organelle membranes, there's a lot of considerations in how that all happens. Dr. Merrill will probably talk some about that next term when he talks about membrane transport. But suffice it to say that moving things across membranes is not a trivial function. So cells use various ways of identifying or flagging where a protein is supposed to go, okay, and how a protein is supposed to function. So some of these are the decorations that we see of the glycoproteins. Some of the things we see in the glycoproteins, for example, serve as identity markers. So when we think, for example, of let's say a kidney, and a kidney has various things associated with it. We think about transplanting kidneys. Okay, we transplant a kidney from one person to another. We can do that with varying success. And when we hear about failure associated with a transplant, we hear of rejection of that. That rejection happens because there are different patterns of markers on different people. So if I want to transplant a kidney, for example, from one person to another person, I want those markers to match as much as I can, so I have less probability that the immune system will see it as foreign and attack it. And that's all happening because of these sort of identity tags that are on the surface of the cells, and these are arising from the glycoproteins, kind of like what we saw with the blood cells and so forth. So these are important considerations. So it's important that we understand then something about how these glycoproteins get made. This is an electron micrograph showing some important cellular organelles. We see the endoplasmic reticulum, which plays a very important role in the synthesis of many of the glycoproteins, and we also see the Golgi apparatus that is also important in this process. So let me just take you through an example modification of some proteins. So what I'm going to be talking about first are the modifications that can happen to N-linked glycoproteins. N-linked glycoproteins start being modified in the endoplasmic reticulum. Okay? They start being modified in the endoplasmic reticulum. They are then transported, usually, to the Golgi, where they may get further modification. So N's start in the endoplasmic reticulum and then move to the Golgi. O-linked glycoproteins start and end in the Golgi. So there's a difference between the two in terms of where their modifications occur. What I'm going to show you now is how the N-linked actually get made, or describe briefly to you how the N-linked ones actually get made in the Golgi apparatus. I'm sorry, in the endoplasmic reticulum. I can't even get it straight. Okay, so what you see on the screen is an odd molecule. It's called dolichol phosphate, and looking at its structure, you can see that it is a long molecule. That's indicated by this N here. N is 15 to 19 of these guys here. You see that this long molecule has a very, very nonpolar end. Okay? Nothing in there to interact with water. At its other end, it has a phosphate. Phosphate, of course, likes to interact with water. Well, it turns out that this molecule like other molecules of its chemical structure, really likes to embed itself in the lipid bilayer of membranes. We find dolichol phosphate in the lipid bilayer of the endoplasmic reticulum. Okay? And it's oriented in an interesting fashion. If there's nothing attached to it, we see the phosphate portion of the molecule pointing outwards from the endoplasmic reticulum. So if we think of the endoplasmic reticulum as being an enclosed organelle, and it's in the cytoplasm, so if we look at back here, we go back to our figure. Okay, we look in the what did I do? When we look at the endoplasmic reticulum, okay, all this stuff out here surrounding the endoplasmic reticulum, all this stuff out here is cytoplasm, right? So the dolichol phosphate is arranged, okay, so that on the outside, or actually here's the endoplasmic reticulum. It's arranged, so here's all cytoplasm out here. This is Golgi up in here. We see that we have an outside portion of the endoplasmic reticulum and we have an inside portion. Outside touches the cytoplasm, inside is the inside part of the organelle. The dolichol phosphate is arranged so that it's on the outside, its phosphate is pointed to the outside to start. It's arranged on the outside to start. And it's onto the outside portion of dolichol phosphate that Y-shaped structure for N-linked glycoproteins is made. So out here on that phosphate, on the outside portion of the lipid bilayer of the endoplasmic reticulum, that common core that we see in N-linked glycoproteins that was Y-shaped, it had N-acetylglucose, and those 3 mannoses. Those get put on to the dolichol phosphate on the outside portion. And then, very interestingly, what happens is the addition of those 3 residues to the phosphate of dolichol phosphate causes, by an unknown mechanism, causes that molecule to flip from being outside to now being inside of the endoplasmic reticulum. So now the Y-shaped structure that was hanging out on the outside of the endoplasmic reticulum is now on the inside of the endoplasmic reticulum. And then it can be attached to a protein in the endoplasmic reticulum. Then it can be attached to the protein in the endoplasmic reticulum. So now we've got that Y-shaped structure on the endoplasmic reticulum. We can start decorating it. We can put other sugars onto it. Some of those will happen in the endoplasmic reticulum. Some of those will happen as it gets moved to the Golgi apparatus. Okay, everybody got that? Yes, Anisio.
As for purification, so the phosphate
Yes.
moves internally
Yep.
to the ER after the combination of the
Mm-hmm.
inhibitor.
Yep.
Throughout the process.
Yes? So what was the question? I'm sorry.
No, I was just asking to clarify.
So it does. So it flips in.
Okay.
It transfers, so that Y-shaped molecule gets transferred to a protein, and then it flips back out. So it's basically a flipping thing. I mean, it's kind of a cool system. And as I said, I don't think much is known about the actual mechanism of the flipping, but it does flip back and forth between the two. Okay. So as I said, that Y-shaped structure can get decorated as appropriate. It can get moved to the Golgi apparatus and be decorated further. Okay. This shows some of that trafficking that's occurring, that can occur in the cell. Here is the endoplasmic reticulum. We see proteins being made and modified in there. We see budding off that can then merge with the Golgi apparatus. The Golgi apparatus where additional modifications can happen. And now those modifications, those modified proteins, now all have little identity tags about where they're supposed to go. Some will get moved to the membrane where they will become embedded in the membrane. Some will get secreted from the cell, as we can see here. And still others will be sent to other organelles in the cell, including here what's called the pre-lysosome, which ultimately becomes the lysosome. The lysosome is a very important cellular organelle that we can think of as the garbage cleaner. All right? Lysosomes are very important in degrading things in the cell. The cell internalizes some nutrients and it needs to break it down. It may use the enzymes in the lysosome to do that. The lysosome is full of degradative enzymes. The lysosome is full of degradative enzymes. So we can imagine that getting those enzymes into there is a very important property, a very important thing to do. If we don't get those enzymes into there properly, we're going to have problems. And in fact, we do. I'm going to lead you talk about a genetic disease in just a second that illustrates that principle. Okay. To tell you that disease, I first of all need to tell you about a modification that has to happen. Okay. So I said the decoration of those N-linked glycoproteins had to happen, and that modification provided an identity tag and basically a destination tag. We can think about it almost like an airline ticket. Where are you going to go with this tag that you've got? Well, one of the modifications that has to happen that tells the cell that this is a protein that's headed for the lysosome is a modification of a specific mannose residue to put a phosphate onto it. Doesn't matter which one, but one of the phosphates in a glycoprotein has to be modified to put a phosphate on position number 6. And we see those reactions occurring. There's a mannose attached to that N-linked glycoprotein. There is an intermediate, and then there is the final product, mannose 6-phosphate. So we see that phosphate being attached. This guy on the bottom with that phosphate attached on it tells the cell, I'm supposed to go to the lysosome. Okay? Now, there are people who have a genetic disease where they lack one of these enzymes. Okay? They lack the ability to make this mannose 6-phosphate. Now, they have a disease called I-cell disease. All right? Now, what's interesting is that these people, because they have this disease, do not get these enzymes moved to the lysosome. They do not get the enzymes moved to the lysosome. They make the enzymes perfectly well. They've got all, in fact, when you look at the people who have this disease, they are loaded with these enzymes. In fact, they make so much of these enzymes, they excrete them in their urine. Okay? Why are they excreting them in the urine? Well, the cell gets so full of them because it doesn't make them into the lysosomes. They start getting dumped out of cells, cells bust open, etc. Okay? It's a very debilitating disease. In fact, it causes severe neurological problems. And it happens because of the lack of that one phosphate on that mannose. So I bring this up to illustrate the importance of those licensing tags that tell where something is supposed to go in the cell and the consequences of what goes wrong if we don't get those things put onto there. Did you have a question?
I just missed the name of it.
It's called I-cell. The letter I, I-cell disease. So I-cell disease happens simply because of that. And as I said, they make perfectly normal proteins. They make abundance of proteins. But because they're not in the lysosomes where they would be functioning to degrade things, they end up wreaking havoc, literally. Okay. Let's see. There's only one other thing I want to talk about, and then I thought we would sing a song. The last thing I want to talk about actually relates to viral receptors down here. And we can see, again, the importance of these glycoproteins. What you see on the screen is a flu virus. And this flu virus is the ball that we see at the top. And at the bottom, we see a host blood cell. So the host blood cell has on its surface, as do all cells, has a glycoprotein. The name of the glycoprotein doesn't really matter for our purposes. But suffice it to say that this flu virus is looking for a specific structure of carbohydrate on a glycoprotein that's found on a blood cell. Okay? The flu virus has two important proteins that are on its exterior. You see the flu virus, it looks like this. It actually has a lipid bilayer. It has multiple RNA molecules inside of it. And on the outside, it has these proteins sticking out like spikes. One of the proteins is called hemagglutinin. And hemagglutinin is the protein that's looking for and binding to this specific carbohydrate structure on the surface of this blood cell. This particular virus has found it, its hemagglutinin has bound to it, and it has attached itself to the blood cell. It's in the process of making an infection. So we're about ready to transmit flu viral RNA into this blood cell. Okay? Well, the attachment alone is not sufficient to get the RNA into the blood cell. Remember that we have to inject the nucleic acid into a cell in order to infect it. In order to get the nucleic acid into this cell, there's actually a cleavage of a sugar in this glycoprotein that's necessary for the entry of the viral RNA. The cleavage of that sugar is catalyzed by the enzyme neuraminidase, which is also on the surface of this flu virus. So once the hemagglutinin attaches itself, you see that right next door, here's the neuraminidase. It's ready to cleave a residue and then inject the viral RNA. If the neuraminidase cannot cleave that sugar residue off, the viral RNA cannot be injected in it. All right? This is a strategy for an anti-flu drug. And in fact, it is the strategy that's used by the anti-flu drug known as Tamiflu. Anybody ever had Tamiflu? Have you used Tamiflu? Any side effects? No? Okay. Did it work? Okay. So Tamiflu is what it does, it's a neuraminidase inhibitor, and it prevents the neuraminidase from functioning. Well, since the neuraminidase can't function, the virus can attach, but it can't inject its viral RNA. Okay? Well, Tamiflu doesn't work against all strains of flu virus, but it's very effective against some and is used quite commonly now to treat certain types of flu that can cause some problems. Okay, that I think is enough for today. I do have a song that's a very fun song. If you'd like to sing it, we can sing it. It's all about sugar structure. And it is called Hark the Sucrose. You can probably figure the song it comes from. And please join me. Carbohydrates all should sing, glory to the Hayworth ring. And oh, merry carbons hide when they're in a glycoside. Glucopyranose is there in the boat or in the chair. Alpha, beta, D, and L, diastereomer hell. Alpha, beta, D, and L, diastereomer hell. In singing that song, it's a good thing I sing the songs because I realized things I didn't say. A Hayworth ring, when you make a ring structure, that's called a Hayworth ring. Okay? The straight chain is called a Fisher structure. So those are two things I should have mentioned. So good thing I brought the song up. Okay, see you guys tomorrow. Yes, ma'am.
I was wondering if you received my, I dropped off the...
Yeah, I got it. I saw it on my desk. Yeah, yeah, sure.
He'll probably still be here tomorrow. Yeah. You never know.
Are you free tomorrow anytime to discuss the exam?
Sure. If you look at my, do you have my calendar link?
Yeah, it's on the syllabus.
Yeah, take a look at my calendar. It'll tell you where I'm at. Okay? But I'll be happy to. Yeah, sure.
Do you have time right now to talk about the test or just...
I promised somebody else I would right now, but if you want to look at my calendar, we can talk about it maybe tomorrow.
Okay.
Okay?
Okay, thank you.
Yes, sir.
Did you upload the eCampus grading distribution?
eCampus grading, I will upload tonight.
Okay.
Yep. Okay? Now, you wanted to talk right now, right?
Yes.
Okay. Let me get everything packed up here, and then we can talk. Just a second.